Hi students, today we are going to discuss about another important 5-mark short notes. Actually this is from OFT, that is organ function test. In this organ function test, now we are going to discuss about the liver function test. Your question is actually laboratory findings in the differential diagnosis of jaundice, right? That means what are all the laboratory findings in all the three types of jaundice? Okay. So first you have to mention three types of jaundice. You all know hemolytic jaundice, hepatic jaundice, and obstructive jaundice. Hepatic jaundice means the problem is in the liver. Hemolytic jaundice means the problem is before liver. Obstructive jaundice means the problem is after liver, that is in the gallbladder, bile duct, like that. So, for these laboratory findings, you have to mention 5 important parameters or points, right? 5 parameters. In blood, actually, serum bilirubin, 2 parameters, conjugated bilirubin and unconjugated bilirubin. You have to mention whether the level of unconjugated or conjugated bilirubin is normal or high or low, right? Like that in urine, you have two parameters, that is urine urobilinogen and urine bilirubin. The last one is fecal urobilinogen. This is very, very important MCQ. Fecal urobilinogen. It is increased or decreased or absent, right? Now, I want to start from obstructive jaundice because most of the university questions are in the practical charts. They preferred, any college, they preferred for their university examinations obstructive jaundice only. So, in obstructive jaundice, what is the result of conjugated and unconjugated bilirubin? You know the conjugation and unconjugation meanings. After RBC hemolysis, the bilirubin is called as unconjugated bilirubin. Then enters into the liver where it will get conjugated. So, in the liver and after liver only conjugated bilirubin. Before liver, unconjugated bilirubin, right? Here the first topic is type is obstructive jaundice. In this obstructive jaundice, there will be no place for unconjugated because after liver. So unconjugated bilirubin is normal, whereas conjugated bilirubin is increased because from the liver, all the conjugated liver enters into the gallbladder and bile duct. So in obstructive jaundice, because of the obstruction, you will get more amount of conjugated bilirubin, whereas unconjugated is either normal or low, right? Second one is hepatocellular problem. Here, hepatocellular jaundice. Here the problem is in the liver. So initially unconjugated is high and after that conjugated is high. So in hepatic jaundice, both conjugated and unconjugated is present, bilirubin. Third one is hemolytic jaundice. Here the problem is hemolysis. So that the conjugation, there is no place for conjugation. So conjugated bilirubin is normal or low in hemolytic jaundice. Whereas unconjugated, this is before liver. Unconjugated bilirubin in hemolytic jaundice is increased. Right? So very simple, don't get confused. Hemolytic jaundice, conjugated bilirubin normal, unconjugated high. Hepatocellular jaundice, both conjugated and unconjugated bilirubin plus. Obstructive jaundice, unconjugated absent or normal or conjugated increased. Okay, this is regarding serum bilirubin. You know the value of bilirubin, actually conjugated is 0.0 to 0.2 and unconjugated is 0.2 to 0.7 milligram per deciliter. You should mention the normal value also. Conjugated is very less, 0.1 to 0.4, some books says 0.0 to 0.2, whereas unconjugated is high, that is 0.2 to 0.7 milligram per deciliter. Okay. Coming to the urine, we have again two parameters, urobilinogen and urine bilirubin. In obstructive jaundice, there will be no place for absent because urobilinogen is the hallmark for hemolytic jaundice only. So, urine bilirubin is present in obstructive jaundice because the liver conjugated bilirubin, bile duct conjugated bilirubin is there. It will get excreted in the urine. So, in obstructive jaundice, conjugated bilirubin, that is urine bilirubin present. Whereas in hemolytic jaundice, urobilinogen is increased, whereas no serum bilirubin. Got it? And in hepatocellular, the urobilinogen is decreased, actually very minimally present. Whereas in urine, the bilirubin is, if micro obstruction is present, obstruction level is very small, that is called micro obstruction, you will get urine bilirubin, otherwise no bilirubin. Okay, this is regarding urine bilirubin. Coming to the fecal urobilinogen, in hemolytic jaundice, the level is so high, that is 40 to 280 is your normal level. 40 to 280 milligram per 24 hours is urobilinogen normal level. Whereas in hemolytic jaundice, it will get markedly elevated. And in hepatocellular jaundice, there will be no hemolysis takes place. So normal or decreased. Whereas in obstructive jaundice, trace. Trace means very minimal. So this is regarding your lab diagnosis in the three types of jaundice, right? So first you have to mention about the conjugated and unconjugated serum bilirubin levels. Then urine urobilinogen. Urobilinogen means first word coming to your mind and brain is hemolysis. So hemolytic jaundice, urobilinogen. So in both urine and fecal urobilinogen, the level is high for hemolytic jaundice. The other two types, no place for the urobilinogen. Whereas in obstructive jaundice, urine bilirubin is present, that too conjugated bilirubin. Urobilinogen is absent in obstructive jaundice, and fecal urobilinogen very trace in obstructive jaundice. Obstructive jaundice is very, very, very important, right? Hepatic jaundice in between, that is initially unconjugated bilirubin. After that, conjugated bilirubin, all the unconjugated bilirubin will get conjugated and converted into conjugated bilirubin in liver. That is why in hepatocellular damage or liver disease, you will get both conjugated and unconjugated bilirubin, right? Normal bilirubin level is very, very important. Total bilirubin you have to mention, unconjugated you have to mention, conjugated bilirubin. Three different bilirubin levels you have to mention for this 5 mark short notes. Okay.